All right, in today's lesson, we're going to be looking at two things: prokaryotic versus eukaryotic cells, and then the eukaryotic organelles within the eukaryotes themselves. So starting off from our textbook, a eukaryotic cell has its own definition, as you can see here, and it kind of is defined in contrast to a prokaryote. So a cell that differs from a prokaryote having a nuclear membrane, which is a nucleus, and then subcellular organelles, which we'll get to next. Also, they're also defined as having mitotic cell division. We're not going to focus on that here. It's a different kind of cell replication. Now, to kind of put that into 2 bullet points, sort of summarizing the definition in a different way, eukaryotes can be described in sort of 2 distinct ways from prokaryotes. Their DNA is surrounded by a double membrane. That is the nucleus here. And then they contain cellular organelles, membrane-bound subcellular organelles from the definition. So kind of taking that definition and parsing it out into 2 bullet points there. Those are the 2 general distinctions that apply at the highest level. Now, organelles are a key part of eukaryotes. It's one of the key features. And what they ultimately allow is internal organization or compartmentalization in a way that is similar to having multiple rooms in your house. Allows you to compartmentalize your house and then sort of perform different activities in different parts of the house. So internal organization, kind of like the rooms of your house. So these different little organized areas are known as the organelles. The term is actually derived from the word organ, kind of like the organs of your body. Each organ specializes as part of the whole. Now, there are a variety of different of them out there. We're going to focus on the most common here towards the end of this video. But basically, an organ an organelle rather is like an organ, a specializing part of the whole. So just like your heart specializes in pumping blood, your lungs specialize in exchanging oxygen, your kidney specializes in detoxification, amongst other things, and each of those can be considered an organ. And just like the organ has a special function, cells can have similar sorts of makeups in which each part of the cell has its own specialized function. So the term organelle is actually derived from the root word organ. We were studying human organs long before we ever even saw cells with a microscope. So we were already familiar with organs before organelles became a thing. So when we look back at the idea of prokaryotes versus eukaryotes, we can sort of summarize key differences here, and then we'll go into more detail in a diagram coming up. But prokaryotes, we can think of them as simpler by comparison to the eukaryotic cells, which are more complex. Prokaryotic cells are not really simple, they're fairly complicated, but compared to eukaryotes, they're less complex. A couple more distinctions: that prokaryotes on average are smaller in size, they lack the organelles, their DNA is unprotected, and not mentioned so far, they do not form multicellular organisms, or put another way, they only exist in a single-celled form. Really, the eukaryotic bullet points here are contrasting. They're larger in size on average, contain the organelles, their DNA is protected by a nucleus, and they're capable of forming multicellular organisms. It is worth pointing out that eukaryotes can be both single or multicellular, so they can be both. Prokaryotes are only single-celled, so they only remain in that one form, single-celled, whereas here we can find examples of both single and multicellular. But you have the ability to be multicellular, whereas in prokaryotic organisms, they cannot do that. Now, as a real quick lesson here on how an organism can become multicellular, cells themselves can group into what are called tissues, which is a collection of specialized cells, or cells that all form a specialized function together, So it's basically one kind of cell all grouping together. If you get 2 or more tissues that join together to serve the same function, we call that an organ. If we get 2 or more organs that group together to form a specialized function, we call that an organ system. And then multiple systems make up the organism as a whole. And this is really more of an anatomy and physiology lesson. Some of you may be familiar with this from anatomy and physiology. Not really going to emphasize this too much, but just to connect it to the idea that this is how you can become multicellular when that applies. You go from cells to tissues, organs, organ systems, up to the organism. So this figure here, these 2 figures, show a eukaryotic cell on the left compared to a prokaryotic cell on the right. I showed this figure last week, and so this figure is to kind of contrast. It's not the best figure in the world, but it's what it's trying to show you is that you have the cell and then the internal organelles shown here that make up these specialized areas of the cell. And also, although this figure here is the same one here, this part being unlabeled, these two are meant to show a size comparison. So this is about the size compared to the size of this cell on average. So this is not actually the same size if you were to look at it from a realistic depiction. So these are just sort of references that you can look back for visuals here. Okay, I'm going to go to the whiteboard here. Hopefully you can see this all right. What we want to look at is the comparison with the diagram of eukaryotic and prokaryotic cells. So what we want to do is in the space provided on your lecture guide there, you can draw out how it works for you, but we want to sort of have this idea of a eukaryotic versus a prokaryotic cell. And we want to start off by just drawing what represents the cell. And we're going to draw these in a general size dynamic so that the eukaryote is going to be roughly 10 times the size of our prokaryote. Although I don't think that exactly is the right ratio, but in terms of size, we're looking at a ratio of 10 to 1. So this would be a comparison of size. This is going to be 10 times larger than the prokaryote. You might want to write that down differently so you don't get that confused later, but that's what that means, 10 to 1 average size difference. So these are 10 times bigger. Okay. Next thing that we want to do here is start off by showing what do they have in common and then what's different about them. So let's start over here with common features. And we're not going to go too deep. These are going to be relatively brief, and actually you should be familiar with these because you've actually seen these before. So the common features are going to be that they both have DNA and RNA, which are the hereditary material of the cell. So RNA, DNA/RNA. They're both going to have a cell membrane, sometimes called a plasma membrane. They are both going to have some kind of cytoplasm, sometimes called cytosol. And they're both going to have ribosomes, which are the protein synthesizers. So DNA, RNA, cell membrane, cytoplasm, and ribosomes. Now, on a prokaryotic cell, we can show the membrane sort of circling the cell. And if you want to label these, you certainly can. Here's your membrane. DNA and RNA, we'll just show the DNA for simplicity, even though they're both there. But the DNA is obviously right here. Cytoplasm is just the fluid, internal fluid. It's always a little difficult to point that out because it just makes up the internal space there. And then ribosomes are going to be shown as these little dots. They're relatively small by comparison, and those ribosomes maybe can be represented as the collection there. So in a eukaryotic or prokaryotic cell, these things are just sort of making up this space around the outside and the inside here, cell membrane being around the outside and the rest of those making up the inside. So those are all pretty minimal features that are common to all life, and prokaryotes will exhibit those 4 features there. Now, as we've talked about from the last lesson, we know that prokaryotes have more features, flagella, fimbriae, etc. But these are just things that allow us to compare them to the eukaryotes. So over here then with the eukaryotes, we're going to see those same features. So eukaryotes are also going to have a cell membrane. They're also going to have DNA. They're going to have cytoplasm throughout the cell. And then they're going to have ribosomes, but for now I'm just going to show those in one area, and I'll talk about why I'm showing them here and not throughout. But they do have those ribosomes. And so this is where I'm diagram this how you want to. I'm going to try to keep this open so I don't draw on top of myself here. But those are the key features there. So membrane, cytoplasm or cytosol, DNA, which includes RNA as well, and then the ribosomes shown here. Okay, so those are the features that you would find in both types of cells. Now what we want to do is look at unique eukaryotic features. So I'm going to diagram these here, and then we're going to go through them in a little bit more detail in the next part of the video. We'll keep it relatively brief overall, but this will give you sort of an outline diagram followed by a little bit more supporting information. So in addition to the basic common features, in a eukaryote, as we mentioned already, you'll have a nucleus, which is what initially surrounds the DNA. Now, there are multiple parts of the nucleus, but for this part, we're just showing that as one big whole. Basically, it's a separate compartment around the DNA. The next what you have is called endoplasmic. So endoplasmic, one word, reticulum. So that's RETICULUM, reticulum. Kind of a crazy or big fancy word there, but endoplasmic reticulum. Endoplasmic reticulum is another sort of separating internal membrane that branches off of the nucleus and sort of spreads out in layers from the nucleus in different areas. I don't have to draw this exactly like I draw it, but you're kind of seeing like a series of layers that sort of branch off of the nucleus, which is this layer here around the DNA. Okay, so endoplasmic reticulum then will show that as being this sort of whole collection here. Now, from there then we can actually divide the endoplasmic reticulum into two subparts, rough and smooth. Rough is abbreviated as rough, ER, rough endoplasmic reticulum, RER. Smooth is abbreviated as SER, smooth endoplasmic reticulum. Now, where I drew these little ribosomes up here, that area represents the RER. And the ribosomes are why they call it rough. Although it's not self-evident, the terminology is based upon someone's subjective opinion. When they looked at it, they said it looks rough. We'll call it rough endoplasmic reticulum. Smooth is therefore contrasting that term. It lacks ribosomes. So areas here where I drew no ribosomes, shown in this area here, that area is considered smooth endoplasmic reticulum. Let me give that just a slightly different contrasting color here, just for a quick contrast here. So the green could represent the smooth endoplasmic reticulum. Okay, so we'll talk about what those do a little bit more here in a second, but those are the where they are located. These are part of the compartments and the organelles here starting from the nucleus. Next one that you'll have is called the Golgi, Golgi apparatus. Golgi apparatus is adjacent to the endoplasmic reticulum and looks like a series of flat little pancakes kind of drawn off to the side from your endoplasmic reticulum. So this over here is the Golgi. Talk about what the Golgi does, but it's part of this system of internal organization. Another key part of a eukaryotic cell, is the mitochondria. You've probably heard of this one. A lot of people are familiar. Mitochondria is the powerhouse of the cell. Mitochondria can vary in the number per cell, but they're often drawn something like this, and they're shown as this sort of anywhere in the cell, not necessarily connected to the other parts here. But mitochondria make energy, and they're a key part of the cell. Now, there are a number of other things that we could talk about, but I'm going to keep it to those there. These are really the most common things that you find in all kinds of cells, and especially animal cells, but really all eukaryotic cells. And so we'll kind of keep it to that short list there. So you can see the common features and then the unique features, and then kind of how those fit into the diagram. And the idea is that everything in a prokaryotic cell exists within the membrane itself. In a eukaryotic cell, we do have the membrane, but then we have internal membranes that make up each one of these organelles. All right, looking at the remainder of this lesson, we'll be going through the eukaryotic organelles and just mostly focusing on those that pertain to animal cells, which would be humans, and going through them relatively briefly here. This class is not going to go a super deep dive on these, but just sort of the general characteristics of these. So the main, one of the main distinguishing features of a eukaryote is the nucleus. And so the nucleus is that separating division around the genetic material, which is your DNA primarily. And so one of the things that it does is sort of preserve the genetic material. There's a lot more you can say about that, what that means, but things can damage the DNA, and by having an extra layer around it, it prevents some of that damage. It's not perfect, but it does prevent damage. The other thing that it does more generally is it controls passage in and out. So here's the nucleus in a cell. You can see the nucleus here, the cell here. There are things that want to go from the cytoplasm into the nucleus or from the nucleus into the cytoplasm. Now, the membrane separates the DNA, but there are little pores within the membrane of the nucleus, and that nucleus then therefore is open to things that can move in and out of the pores. Now, these are somewhat selective, so they don't just allow anything, but the pores facilitate transport in and out of the nucleus. So the nucleus basically has the envelope around the outside, the pores that allow little openings, and then a dense area called the nucleolus, and then the rest of it is filled up with chromatin, which is a form of DNA. So you have, those are some of the main structural components to it here, and these are the two main purposes. The next organelle is the mitochondria. Mitochondria, which is actually plural. Mitochondrium would be like singular. So mitochondria, plural, are eukaryotic organelles that specialize in making energy. These are often referred to as the quote-unquote powerhouse of the cell. So energy occurs in the form of ATP. That's the specific form of energy that is made. And so this type of energy is a cellular energy that is utilized by many different parts of the cell. Now, there are other ways that energy can be made, but most of the energy in your body, in your cells, is in this form called ATP. So a lot more to be said about that one as well, but for this class, we'll keep it relatively simple, and we will come back to this in a little more detail later, but mitochondria for now is just going to be the powerhouse of the cell. Energy production. Now, the other thing that's interesting to know is that different cells in the human body have very different amounts of mitochondria. Very often, going back here, you'll see that mitochondria are often shown as a few per cell, sometimes only one per cell. But in reality, some of your cells can have literally thousands of mitochondria per cell. So the more energy a cell requires, the more mitochondria that you'll need. So some examples would be muscle cells, one of the most heavily concentrated. So muscle cells obviously need lots of energy, and they require often thousands of mitochondria per muscle cell. And they're scattered along the length of muscle cells, but very heavily concentrated. It's also true for your liver cells for different reasons. Liver cells require lots of energy, as well as nerve cells that are constantly sending impulses and transmission signals. So those are all examples in the human body that have high energy requirements. Now, on the other hand, some cells require very few mitochondria and therefore very little energy. An example would be on the outside of your skin. You have a skin cell type called a keratinocyte. And a keratinocyte, depending on what stage of development it is, may actually be dead on the very outer layer of your skin or dying as it progresses towards the outer layer. And so these cells are more of a barrier. They don't really have a strong biological function other than to make up the barrier on the outside of your body. So they tend not to use much energy, therefore they don't have many mitochondria. Fat cells are a little more variable, but your white fat, which is what you typically think of as fat, does not require a lot of energy. Now, the exception there is brown fat, and I won't get into the details here, but there's white fat and brown fat. So if you're familiar with that difference, then you might appreciate the fact that brown fat is actually different, and it does have a lot of mitochondria. But what we typically think of as fat is actually called white fat or white adipose tissue, and that has very few mitochondria, relatively speaking. And then the last one here actually has no mitochondria, and these are your red blood cells. Red blood cells are highly modified. They're different in many, many different ways. But one of the things that's different is that they don't really produce a lot of energy, and they don't require any mitochondria. They actually produce a small amount of energy through other processes, but no mitochondria. And by the way, your blood cells are on a timer. Once they're formed, they only last a matter of weeks or months, and then they have to be broken down and recycled into a new cell. So red blood cells don't function like most of your cells. They're on a sort of a short timer, and they only last as long as that timer is good for, which is weeks to months. All right, moving on here to the next one. The next one is more of an elaborate concept. This one's called the endomembrane system. So membrane implies that we're looking at similar features to the cell membrane. Endo means inside, and system implies that there's multiple parts interacting here. So the entire system is made up of different parts that I'll break down next. But overall, what we end up with is a system that's designed for synthesizing and modifying. Kind of like a factory where you build something in its raw form, and then you kind of modify it to get it into its final product form. Maybe that includes painting or sanding or cutting or labeling, adding things together. So you make something and you kind of refine it and then you're ready to sell it. That's kind of how the cell works. You kind of make things in a raw form and then you modify them into their final form. That's one of the biggest generalizations of the endomembrane system. You're making and modifying. Now, things that are synthesized and modified then have to be transported. So transportation is another generalization. So what is made is then delivered. And more variably, and not quite as straightforward here, within the system you have a variety of metabolic and detoxification-like processes. So what you're going to end up doing is creating energy or energy byproducts, really, precursors maybe is a better word for it. We said the mitochondria makes the energy, but the way that works is more complex. You have to feed things into the mitochondria. So some of the precursors will come from the endomembrane system. Metabolic activities kind of feed into the mitochondria, for example, but also breakdown of toxins. So your body is constantly being exposed to toxins, and in order to keep those from becoming a burden or an overwhelming burden, you break them down little by little throughout this endomembrane system. So some of the specific components of the endomembrane system, it actually begins with the nuclear envelope, which is the surrounding portion of the nucleus. And just to jump back here just really quickly, and just to reiterate what I showed here, if this is the nucleus, then this specific portion here, which is the envelope, that actually is where the endomembrane system begins. Okay? And so it begins in that area, and in fact in this diagram you can see this is representing the nucleus here. Okay, and then it transitions into these additional parts, and I'll come back to this same diagram here, but it transitions into different areas. So the first transition point is you go from the envelope to the endoplasmic reticulum, and I mentioned that one earlier in the diagram between a prokaryote and a eukaryote. So going back over that one here with a little more detail, the rough area is considered rough due to ribosomes. Ribosomes are what make it look rough, and that's a arbitrary descriptor that someone came up with, but they call it rough. And since ribosomes make proteins, then that ends up implying and outright stating that the endoplasmic reticulum is designed to hold ribosomes and make proteins. So the combination of the ribosomes in that area is what gives you the protein synthesis. So the smooth endoplasmic reticulum then is somewhat defined by the lack of ribosomes, and that's where the term smooth comes from. It ends up being a lot more complex to describe, but the simple explanation is that it makes lipids and detoxifies harmful substances. What lipids and what harmful substances and what percentage of what is doing in what cell is very difficult to explain. But for example, in the reproductive cells of men and women, lipids will be specialized, synthesis of lipids will be specialized for things like hormones, testosterone and estrogen, for example. So in certain cells, most of the work is on making lipids. Then if you look at your liver cells, most of the work in the liver is actually focused on detoxification. So what they're doing is often more emphasized in one cell type than another, for example. So here I have that example with the specialized cells in the liver for detoxification and reproductive organs to produce hormones. So there's a lot going on there with the smooth endoplasmic reticulum, highly variable from one cell type to the next. But those are the generalizations, and the rough endoplasmic reticulum is much more general. It makes proteins. Now, the next part of the endomembrane system is called the Golgi apparatus. And to give you an idea of how it works, it's often referred to as the post office, quote unquote, of the cell. So if you think of the post office, you take a package to the post office, and they sort of put their own special label on it. Even though you have a label, they put their own scanning label on it, usually. and then they deliver it from the drop-off point to wherever the address tells them to take it. So they basically send and receive packages. And in that regard, the Golgi apparatus is called the post office because it turns out it does something very similar. So it's involved in transportation and secretion, and what it basically does is it takes products from one part of the cell and transports them into another part of the cell. So I'll go back to the diagram here so you can see that, give you a chance to write this down, and then I'll come back to the nerve cell as an example here. So let's just go back real quickly here to this diagram. So here you can see that this is the nucleus, this is the rough endoplasmic reticulum, this is the smooth endoplasmic reticulum. So proteins and lipids are being synthesized here, and then what I'll get to last is they're packaged up in little vesicles that literally move from one part of the cell to the next. And this over here is the Golgi apparatus. These are flattened sacs that all consist collectively of the Golgi apparatus. Individually, these are called Golgi bodies. If you want to refer to each layer, you can call it a Golgi body. But collectively, it's the Golgi apparatus. And as you can see from this diagram, there's little proteins, these little red dots, or sorry, green dots here. Those represent proteins. They're being packaged up in this little circular structure. And then they're showing that circular structure moving over here from the rough to the Golgi apparatus. And now those little proteins are being fused into the actual Golgi. And then from there, they'll migrate out the other side, one direction or the other. And then that will literally represent the point at which they're transported to where they're supposed to go. So imagine this is the point of origin for the package. You're driving the package to the post office or the point where they're going to pick it up. They sort of put a little special label on it, make sure they know how to process it, and then from there it leaves the post office and it goes wherever it's supposed to go. And sometimes that means it gets dumped to the exterior of the cell, and sometimes that means that it comes back into the cell and is used internally. But that's the Golgi apparatus in a nutshell there. It transports and secretes substances, and where this plays a role, I mean, it's all of your cells, but the nerve system and nerve cells in particular is an interesting area where this is especially important. Neurotransmitters are the products that are being produced, and then the secretion of the neurotransmitters is the process by which the nerve cells communicate. So they'll make the transmitter and then dump it to the outside of the cell, and that actually acts as a signaling molecule which causes other nerve cells to fire in response to the neurotransmitter. That's a whole separate lesson. I don't want to turn this into a lesson on the nervous system, but as an example of where the Golgi bodies play a key role is in the function of nerve cells and therefore the nerve system. And the last thing here is that the products that are being transported are going to be held in generalized structures called transport vesicles. So I'll come back to this one here. This is that same figure here labeled, by the way. And these little structures here are transport vesicles, as it says. So those transport vesicles are going to transport substances from the endoplasmic reticulum to the Golgi apparatus. Could be anything, but that's the idea. Whatever's in there is different from cell to cell, even moment to moment, but that's a generalization. Now, in animal cells, those transport vesicles become modified and can end up doing things that are sort of unique. So in the animal cell, some of these vesicles will grow to a larger size and they'll develop into specialized forms called lysosomes. So a lysosome is a specialized sort of vesicle that contains enzymes to break down cellular components. Cells need to sometimes be broken down, and sometimes cells eat other cells. So for example, your white blood cells will eat bacteria, and then they'll use the lysosomes to break down the cell walls of the bacteria, for example. So lysosomes are like the stomach of a cell. They'll really break down a lot of the big chunky stuff, and it helps them turn it into smaller bits for recycling. And then peroxisomes are similar. They contain digestive enzymes as well, but they tend to be more specialized for food products, things like the proteins, carbohydrates, lipids, and others. And there's a lot of variation as to how these work in different individual cells, but those are some generalizations there. These are really specific to animal cells. So lysosomes for large cellular material and peroxisomes more for the major food groups, but others as well there. So inside the cell, then, this endomembrane system really represents a large part of what's happening in a eukaryotic cell. Information from the nucleus is passed into the factory, if you will, for synthesizing proteins and lipids. The products in their raw form are packaged and taken to the Golgi and modified and then repackaged for delivery to where those products are going, inside or outside the cell. So that really represents one of the main areas where eukaryotic cells specialize, is internal organization and the flow of products and the dissemination of those products in a highly organized fashion. Although it's hard to connect these dots here, that ultimately allows these cells from the cell level to become more complex in multicellular forms. Without these internal cell complexities, a cell can never actually organize itself to the point that it could become multicellular. So these are all key developmental aspects that lead to even more complex aspects like multicellularity later down the road. All right, we'll